Joss. Hello, my name is Agustin Luján. Everyone call me Agu, and I'm joining today from Barcelona, Spain. I'm a TANGO2 clinician researcher at the Hospital Sant Joan de Déu in Barcelona, and a member of the research committee of the TANGO2 Foundation. And I would like to introduce our next speaker, Dr. Michael Sacher. Dr. Sacher is a researcher at Concordia University in Canada and currently serves as a member of the TANGO2 Research Foundation Board and the Scientific Advisory Board. His lab has established several TANGO2 model systems, including the fruit flies and the human fibroblasts, to aid in their work. Their experimental studies confirm Dr. Miyake's hypothesis that B vitamins are essential for TDD, but unfortunately, Dr. Sacher is unable to join us live and has pre-recorded his presentation.
So thank you. I appreciate the invitation to speak today, and my apologies for not being able to present live. I have teaching responsibilities on Fridays at 10 a.m. and a time that was working for everyone could not be scheduled. So my talk is bread, beer, and rare disease. Why did I choose that? Well, it turns out that bread and beer share a common ingredient, which is yeast. And I'm going to talk today about our use of the yeast model system to study TANGO2, to study the TANGO2 homolog, Tng2, as well as to study the human TANGO2, which you'll see how that evolves as we go along. I'm not going to give any background to TANGO2 or to TANGO2 Deficiency Disorder to this audience. I think that is not necessary at this time. Others may give the background for that. Instead, what I'm going to do is focus my talk on model systems and the power in such model systems. So first, what I want to do is define what is meant by a model system. And for that, I'm going to define it as either a whole organism, a cell, or some biological entity that will allow us to study a protein involved in a biological process, or to study just the biological process itself without any particular protein in mind. Now, when we're talking about a human disorder, then the model system that one would ideally like to work with is the human. And that is in fact done. We can perform clinical observations on how this disorder presents itself. There can be natural history studies, and in the case of TANGO2, there is an ongoing natural history study that everyone should be involved in. But when it comes to doing experimentation on humans, well, we run into a bit of an ethical dilemma over there. But that's not a problem, because we can derive cells from humans. So we can extract the easiest cells to get are skin cells, or what we call fibroblasts, right at the surface. Slightly painful procedure, but readily accessible. Want to go a little deeper, maybe a little bit more invasive, so we can do biopsies on things such as muscle cells. Okay, but what if we're talking about a disease or a disorder that is affecting something other than skin and muscle? Maybe it's affecting heart. Maybe it's affecting nervous tissue. How can we work with those? We can't very easily biopsy those. But not to fear, we can take the fibroblasts and we can reprogram them. Now think of fibroblasts, they're a specialized cell. We say that they're differentiated. You can think of them as being different from other cells. They express different sets of genes. They have different tasks than other types of cells. But what we can do is de-differentiate them or reprogram the cells. And in that case, they turn into what we call induced pluripotent stem cells. And now if you take these iPS cells and you treat them with certain factors, we can get them to differentiate now into cardiac cells or adipocytes or into neural cells, nervous tissue. And these might be better model systems to study than would be a skin cell or a muscle cell. Okay, so the drawback to this, however, is that it requires very specialized work, and it is very expensive to perform this work. The reagents that are necessary to differentiate the cells, the growth conditions, it's not easy to work with these iPS cells at all. Okay, so we can have another model system, mouse, right? Everybody has heard of mice being used in experiments. There's, of course, reduced ethical considerations in the use of mice compared to the use of human cells. They do have some drawbacks as well. Now, the results can be affected by a variety of environmental factors. And so depending on how the mice are housed and treated, different results can be obtained by different laboratories. They don't always replicate human conditions. And there is a cost involved in housing the mice, breeding the mice, feeding the mice. Okay, but these model systems that I've discussed up until now, humans, mice, cells derived from those systems, those are mammalian model systems. Right? Those are model systems that I would say your non-basic researcher is much more comfortable with. But outside of basic researchers and maybe some clinician scientists, the power of non-mammalian model systems, I think, is lost on some people. And I'm going to focus on that today. So just as a sample of non-mammalian model systems that can be used for rare disease research, we've got the fruit fly, the frog, the worm, C. elegans. You may have already heard a talk from someone in Dr. McKenzie's lab on how they're using C. elegans, or from Dr. Gupta's lab on the zebrafish model system. I'm going to talk today about the use of yeast. And I'm going to refresh your memory on how we actually already did some work on fruit fly and sort of try to tie those two together in a way. So again, some of you who don't work with non-mammalian model systems may be asking yourselves, what benefit can we derive from yeast? It's a single-celled eukaryotic system. All these others to the left over here, flies, frogs, worms, zebrafish, these are whole animal models. What could we possibly learn from a single-celled eukaryote? But I would like to remind you that TANGO2 is a highly conserved protein, all the way down to what one might consider the lowest form of life, that being a bacteria, a prokaryotic cell with no internal compartments. I think we're going to see that the answer as to what can be derived from a single-celled eukaryote is potentially a lot. But I would like to just put up these names before I get into the data. These might not be familiar to many of you, may not be familiar to any of you. Randy Sheckman, Leland Hartwell, Paul Nurse, Yoshinori Osumi. These researchers have two things in common. They are all Nobel laureates, and as you might be able to guess where I'm going with this, the Nobel Prize-worthy work that they did was using yeast. As long as you're looking at a protein that is highly conserved or a cellular process that is highly conserved, using yeast is an excellent way to study that process. Now, this slide has been taken directly from my talk at the family conference in the summer of 2022, when I was first introducing our use of the fruit fly model and our work with vitamin B5. And I asked at the time, what can a fruit fly possibly tell us about TANGO2? And as it turns out, the fruit fly told us a whole heck of a lot. And I'm not going to go through all that data, but I'm just going to give you a little sampling of what we did. For those who may not recall, for those who may not have been present in 2022. We looked at a TANGO2 mutant, that's represented by TM, and control flies with a wild-type TANGO2 gene. And first thing we did was we subjected them to starvation. We removed the food and asked how long they would survive for. The controls are shown in black, and the mutants are shown in blue. And you can see that the TANGO2 mutant flies died much sooner during starvation than did the controls. We looked at seizures. We can induce seizures in the flies by raising the temperature. In our case, the control flies did not experience seizures, but the TANGO2 mutants, about 60% of them did experience seizures. We looked at the ability of the flies to learn to avoid a well-lit area that has something bitter in it. Flies like sweet things. They also like the light. They have to make a decision between those two in our little experiment, and the controls were able to make that decision and avoid the bitter, well-lit area compared to the TANGO2 mutants. So this is just a sampling. We did a lot more experiments, and we were able to show that the flies indeed replicate many of the phenotypes that are associated with the loss or dysfunction of TANGO2. And importantly, and for reasons that I don't have time to get into why we were looking at vitamin B5, all of these phenotypes and the ones that we're not showing were rescued by inclusion of vitamin B5. So the TANGO2 mutants with vitamin B5 included is in green, and you can see how that curve is now shifting much closer to the control curve during starvation. You can see how there is a stark reduction in the number of flies experiencing a seizure when they are pre-treated with vitamin B5. And the flies that are treated with vitamin B5, the mutant flies, show a significant increase in their ability to learn. Okay, so there was a lot that we could derive from that non-mammalian model system. Well, the question is, was the vitamin B5 effect that we had identified, was it translatable to humans? And the answer is a resounding yes. So we were able to show a membrane trafficking experiment where in the absence of TANGO2 there's a defect, but when we treated those cells, those human cells, with vitamin B5, we were able to rescue that membrane trafficking defect. We later looked at lipids in the absence of TANGO2 in human cells, and we showed that there was a profound lipid imbalance in cells devoid of TANGO2. But if those cells were treated with vitamin B5, we were able to rescue that lipid imbalance. We know anecdotally, and you all know anecdotally, that vitamin B5 is having an extremely good benefit, a very positive benefit on the TANGO2 warriors who are taking it. And in fact, there are publications now where only vitamin B5 has been used to alleviate metabolic crises. And so I think it's clear that the work that we had done on the fruit flies was very translatable to humans. Again, that's because the protein is so highly conserved that I think when we study it in a non-mammalian model system, we're going to learn something that will inform us about the human condition. Okay, so if I go back to this slide that I showed a couple years ago, this year I'm going to change the slide into this. What can yeast possibly tell us about TANGO2? And I hope to be able to convince you over the next few minutes that, like the fruit flies, we can set ourselves up for a very powerful model system that will inform us greatly about function of TANGO2 and perhaps down the road even allow us to screen for small molecules that can help treat TDD. Before I get into it, I'd just like to show you that, or explain to you that the TANGO2 homolog in yeast has not been characterized. And non-characterized yeast open reading frame, yeast genes, are given a standardized name, the Y, just to tell us it's from yeast. The second letter, G, is the seventh letter of the alphabet. It tells us that this gene is on the seventh, is on chromosome 7. The R could also be an L. So the third letter is either an R or L. It's telling us the right arm or the left arm of chromosome 7. And then each one has a three-digit number followed by a W or a C. Why a W or C? Well, James Watson and Francis Crick discovered DNA. And in yeast, one strand is referred to as the Watson strand. One is referred to as the Crick strand. So this is telling us exactly where the uncharacterized open reading frame is. We're characterizing it now. We haven't published it yet, but we're changing the name to TNG2, I think for obvious reasons why we selected TNG2. Hasn't been published. If someone has a recommendation for a different yeast name, they have to have 3 letters followed by a number. I would certainly be open to hearing any other suggestions. Okay, so the first thing we did is we wanted to know where is this TNG2 protein in yeast cells. And so what we did was we put a fluorescent protein called GFP onto the C terminus of the protein. You might think of it as the end of the protein, or at the amino terminus, or you could think of it as the beginning of the protein. When we looked at cells that had it at the end of the protein, you can kind of make out the cells here, but there's nothing really interesting to write home about over here. But when we put it at the amino terminus of the yeast TNG2 protein, now we're starting to see these puncta. This is not mitochondria. Mitochondria look different than this in yeast. It's not the nucleus. We started thinking maybe it's the peroxisome. The peroxisome, for proteins to localize there, will have one of 2 so-called peroxisomal targeting sequences, or a PTS. One of the peroxisomal targeting sequences is a 3 amino acid sequence at the C terminus of the protein, or the end of the protein, and it is an SKL 3 amino acid sequence. What those amino acids are, not important. But the canonical sequence for a peroxisomal targeting sequence, one of the peroxisomal targeting sequences, is SKL at the C terminus. When we looked at yeast, that's Saccharomyces cerevisiae, this is the C terminus of 4 different TANGO2 homologs. This one is from prokaryotes. Okay, so it's a well-conserved protein. Here's from mouse and human. You can see that the yeast has an FKL sequence. That's interesting. Not canonical, but close. The canonical SKL sequence is also preceded by lysines and arginines just upstream of that sequence. Those are, lysines are K, arginines are R, and you can see that 3 of the 4 amino acids preceding the FKL sequence are indeed lysines and arginines. This really made us believe that this is a non-canonical peroxisomal targeting sequence. And so in order to prove that, I go back to this experiment. We now took GFP TANGO2, where the GFP is at the amino terminus, because we need to leave the carboxy terminus free. The last 3 amino acids in the protein have to be SKL. If we put GFP there, like in the case here on the left, we're blocking that SKL. An internal SKL or FKL sequence in our case won't work. What we did here was we simply deleted those last 3 amino acids and we lose these puncta, which is further proof that these puncta represent peroxisomes. And just to show that a little bit more convincingly, we looked at a peroxisomal protein. Many peroxisomal proteins have the name PEX followed by a number. We looked at PEX3 with a particular fluorescent tag on it called Dudre. Here is what's we're showing here is about 10 yeast cells. This is the fluorescence we get for GFP TANGO2, those puncta again. This is the fluorescence we get for the PEX3 protein, and when we overlay them in white, we see where they co-localize. And so this is showing us much more convincingly that TANGO2 in yeast is localizing to peroxisomes. Now, you might be familiar with previous work, and you're going to say to yourselves, well, 1 second there, Dr. Sasher, your lab, as well as other labs, have shown that the human protein associates with mitochondria. So what's going on here? Maybe I don't want to believe this non-mammalian model system where this protein is now localizing to the peroxisomes. But let me inform you about something that you might not be familiar with. Humans perform lipid metabolism, and I'm going to refer to lipid metabolism as beta oxidation, the breakdown of lipids to smaller metabolites that can be used to produce energy. That lipid metabolism, as well as the energy production, takes place in the mitochondria in humans. Yeast, I think, have done us a massive favor. They've separated those two. Beta oxidation, lipid metabolism, exclusively takes place in the peroxisomes. Energy production using the metabolites from beta oxidation, those metabolites are transported to the mitochondria, and so energy production still takes place in the mitochondria, but beta oxidation has moved to another organelle. Okay, now consider that TANGO2 is a very highly conserved, evolutionarily conserved protein. And so the difference in the location of TANGO2 in these two systems, in humans and in yeast, is likely due to evolutionary differences in the organisms, how they compartmentalize these different activities, with one being in peroxisomes, the other in mitochondria in yeast, both in mitochondria in humans. And so there's no need for a peroxisomal targeting sequence on the human protein if it doesn't need to be in the peroxisomes. Okay, but it likely does not reflect functional differences. And I'm going to get back to this in just a few moments and show you that there is no functional difference in human TANGO2 and yeast TANGO2. But to explore the peroxisomal localization a little bit more, we asked, okay, if it really is in peroxisomes, which it appears to be, there are two main transporters or receptors for an SKL or an FKL sequence, those being PEX5 and PEX9. So what we did was we put our GFP TANGO2, leaving our carboxy terminus FKL sequence free at the very end of the protein. We put it either into a wild type background where we see the puncta, and then we also put it into a background where we knocked out PEX5 or PEX9. And it's quantified down here, but you can see the clear difference here. We retain the puncta in the PEX5 knockout, but in the PEX9 knockout, we don't, suggesting that TANGO2 is transported into peroxisomes using the PEX9 system. We wanted to know if there were any phenotypes associated with a TANGO2, yeast TANGO2 knockout. And so standardly, the cells are grown in the presence of 2% glucose. That's considered a rich source of glucose. We reduced it greatly to 0.1% glucose, and you can see here that the TANGO2 knockout in reduced glucose, when the cells are going to be requiring lipids for metabolism, for energy source, you can see a growth defect over here. These are just dilutions, more concentrated, a tenfold dilution, another tenfold dilution. Okay, so from top to bottom, it's the same strain, just more diluted, so you can see a little bit better. The colonies here are much smaller than the colonies in the rescued strain or in the wild type strain. And similarly, in the presence of oleic acid, which really forces the cells to use peroxisomes to generate energy, oleate is a fatty acid. This again highlights the fact that TANGO2 functions in peroxisomes because the knockout doesn't grow at all compared to the rescued strain or the wild type strain. So again, further evidence that TANGO2 is functioning in the peroxisome. Now, interestingly, if we humanize those yeast, and what do I mean by humanization of yeast? Well, what we did was we deleted the gene encoding TANGO2, the yeast TANGO2, and replaced it with the open reading frame encoding human TANGO2. And wouldn't you know, if we look at where we have our growth phenotypes, 0.1% glucose, the TANGO2 knockout doesn't grow. But if we replace that TANGO2 gene with the human TANGO2, it grows. And look at oleate, where this strain doesn't grow at all in the absence of yeast TANGO2, but when we've humanized it and put human TANGO2 in, now it grows. And this proves that the function of TANGO2, human TANGO2, is conserved all the way down to yeast, further strengthening the argument that whatever we derive from the yeast model system should be translatable to the human system, very similar to what we did with the fly system, where we discovered vitamin B5 had beneficial effects. I'm going to come back to this humanization in just a few minutes. Okay, but I come back to this point that I made before, that the difference in location, right, with human TANGO2 being in mitochondria, yeast TANGO2 being in the peroxisomes, is likely due to the evolutionary differences in how these organisms are compartmentalizing certain functions. Beta oxidation is taking place in peroxisomes, in the yeast, in mitochondria, in human cells, and it likely does not reflect any functional differences. And this strongly suggests to us that TANGO2 has a function in beta oxidation, either upstream of beta oxidation, making it more efficient, or maybe downstream of beta oxidation, where it might be able to shuttle some metabolites to the energy-producing compartment, the mitochondria. That's something that we're still working on. We have some interesting data on that, which is not developed enough to present yet today. But we wanted to know, under conditions where lipids are going to be required for energy, does TANGO2 accumulate in the peroxisomes? Well, those conditions are under low glucose, like I've shown you, and oleate conditions. And so what we did was we looked at the fluorescence intensity. We see those puncta, they have a certain intensity, and we can measure that. And so each dot here represents a different experiment. So we did it in triplicate with, I think it was anywhere from 25 to 50 cells. I don't recall the exact number. We normalized the fluorescence in rich glucose containing medium to an arbitrary value of 1. And you can see that in both cases, when lipid metabolism is required to generate energy, there is a statistically significant increase in the fluorescence intensity of TANGO2 in the peroxisome, suggesting that TANGO2 is required in the peroxisomes for some aspect of beta oxidation. So where are we headed with this work now? What I'm showing you here is a plate. It's 384 yeast colonies. Each dot here is a TANGO2 knockout, yeast TANGO2 knockout that has been transformed with one of the 6,000 yeast genes. Okay, so this spot over here has one gene. This has gene number 2, gene number 3, gene number 4, etc. We have about 20 such plates where our TANGO2 knockout has been transformed with a single yeast gene on a plasmid that allows overexpression of that particular gene. And so what we're going to do now is a genome-wide screen where we're going to ask which yeast genes, when overexpressed, can rescue the TANGO2 growth defects that we see on low glucose and oleate. These are experiments that can very easily be done with robotics, and we're just going to pin them from one plate onto two other plates, low glucose, oleic acid, and we're going to see very few colonies probably growing, but we'll know exactly what gene it is because I could tell you exactly what gene this is, or exactly what gene this is, or go to plate number 15 and I can tell you what's in this position here. Okay, so this is a very powerful way of screening for suppressors and to look for the genetic network that TANGO2 functions in. But what do I mean over here when I'm saying we can screen humanized yeast for small molecules? Well, what I want to show you here is some very recent data that still has to be repeated. But what we've done is we've taken a number of the missense variants, including the most common, G154R, in the human TANGO2. And so we've put the wild-type TANGO2 gene into the yeast. We've humanized it, got rid of the yeast TANGO2, put the human in. And then we put in each of the these 5 missense variants that I'm showing you here. And look on low glucose over here. 2 of them, including G154R, show a growth phenotype, much less growth if you just look at the colony sizes here. And on oleic acid, you can see that neither of these is allowing for growth. So now we've got an interesting scenario where we've got humanized yeast containing, for example, G154R, which grows fine in the presence of glucose, but on oleic acid or low glucose, it doesn't grow. We could now take that strain and screen it against a library of small molecules now to find out which ones now allow these strains, this mutant strain, to grow better. Which ones can rescue the function of human TANGO2. We're not even talking about yeast TANGO2 now. We're using the yeast system to be able to set up a small molecule screen over here. Okay, we can then end up with small molecules that rescue a human mutation, not a yeast mutation. Okay, I just want to make that very clear. We're now, the only source of TANGO2 in this strain is human TANGO2 with a mutation that doesn't allow the yeast to grow properly. So that small molecule, when we screen tens or hundreds of thousands of them, we might end up with a couple that allow it to grow. And it's allowing human TANGO2, which I've already shown you, can function properly in yeast. I'm showing you right over here. And I'm showing you right over here that they're growing just fine in yeast. Okay, so this is an amazing system, I think, to be able to perform a small molecule screen. So just to conclude over here, I've shown you that the yeast TANGO2 is in peroxisomes, and that peroxisomal localization increases during conditions where lipids are going to be more heavily metabolized for energy. We're thinking that it's affecting beta oxidation. Upstream, downstream, don't really know yet. Have some interesting ideas though. And as I just recently, just a second ago, talked about genetic suppressor screens in yeast, using the yeast TANGO2 knockout will give us genetic networks that TANGO2 functions in, but we can also take our humanized yeast and perform small molecule screens to try and identify new therapeutics, that treatments that can potentially be used. So hopefully families can appreciate the power of this model system. I think basic researchers and some clinician scientists can see where we're headed with this work. And clinicians who are unfamiliar with the benefits of a non-mammalian model system, I hope now can better appreciate how these systems have been used and are continuing to be used as powerful tools in TANGO2 research. The work on the yeast TANGO2 has been done exclusively by Alia Naz, a PhD student in my laboratory. The 20 plates that we have with all of the individual yeast genes has been done in collaboration with Charlie Boone's lab at the University of Toronto. Funding for this work has come from NSERC in Canada, a chair stipend in Concordia University, and a small grant from the RDMM network, Rare Diseases, Models and Mechanisms, here in Canada. Unfortunately, I couldn't be here to answer any questions. However, here's my email address, and I answer emails very quickly, and I would really appreciate if you have any questions whatsoever, don't hesitate. Please send me an email. If you don't catch this, you can just scan this QR code, which takes you to my lab website, and you'll be able to find my email address there as well. I appreciate the invitation. I hope I haven't gone too long, and I look forward to being able to speak live the next time there's a symposium or a family conference.